So tonight we're going to be talking about one of the most exciting new areas of cancer research, and that is immunotherapy, one that many researchers believe really has a potential to truly transform cancer treatment and lead to long-term control of the disease. We're fortunate to have an accomplished panel assembled today who are recognized leaders in the field of immunotherapy. And let me give a brief introduction here. They're recognized leaders not only here at Memorial Sloan Kettering, but of course also worldwide. To my far right is Dr. Jed Wolchuk. He's the chief of the Melanoma and Immunotherapeutic Service and an accomplished researcher and clinician whose work has completely changed the treatment of patients with advanced melanoma, a disease which just 5 years ago had very few options. His work here at Memorial Sloan Kettering led to the FDA approval of the first immunotherapy drugs for the treatment of metastatic melanoma. In the center, one of Dr. Wolchuk's key research partners is Timothy Chan. Dr. Chan is a director of the Translational Oncology Division. His work focuses on the genomic underpinnings of cancer and is helping to unlock the mystery around why certain patients respond to immunotherapy treatments and others do not. And finally, to my immediate right is Dr. Rainier Brenchens, the director of Cellular Therapeutics, who has led the charge in some of the most interesting and talked about research in the field to date called CAR T-cell therapy. This is an immune system approach that Dr. Brenchens will explain more about in a short while. Early hints at the immune system could be recruited to fight cancer dates back, believe it or not, to the 1800s, right here at Memorial Sloan Kettering. Now let me play a short clip from the recent documentary, Cancer, the Emperor of All Maladies. It shows a little bit of that history.
The idea that the immune system might effectively treat cancer is not new. It was first explored by a 19th century surgeon, William Coley. While working at Memorial Hospital in New York City, Coley came across a strange case, a patient whose tumor seemed to vanish of its own accord after a serious infection.
Coley thought, there's a power here. We don't know quite how to harness it, but I'm going to try to infect people who may have this kind of dire, inoperable form of cancer and see if we can do anything about it. And that, in a messy and unscientific way, was the birth of kind of cancer immunotherapy.
Coley made a compound of infectious bacteria and injected it directly into the tumors of patients, hoping to trigger a fever that would somehow overwhelm the cancer. Some of his patients were cured, but no one was able to duplicate his results. And for decades, immunotherapy remained on the fringes of cancer research.
We have roughly 37 trillion cells in our bodies that undergo billions of cell divisions daily. And with all this cell division going on, there's bound to be some mistakes. Some of those cells will have just the right combination of mistakes or mutations to become cancerous, or a cell has been exposed to a trigger in the environment that has transformed it into a cancer cell. So the idea behind immunotherapy is to wake up the immune system so that our own bodies can fight the cancer. It's a fundamental shift in the way we look at cancer therapy, as explained in this clip from one of our panelists, Dr. Wolchuk.
Cancer is a dynamic interplay between patient and tumor. And for many decades, we have concentrated all of our efforts on the tumor part of that interplay. And now I think we recognize that there is a role to be played by focusing on the patient and how this host immune response can be tuned to try and control the cancer.
So now let's begin our discussion by asking each one of our panelists a little quick question to get us started. And Dr. Wolchuk, let's start with you. Tell us why this is so hot and a little bit about what it really is.
Rather than directly treating the cancer with targeted therapies or chemotherapies or radiation therapy, we are assuming that the patient's immune system, in some cases, can see the cancer cells as being something different or dangerous. And they are trying to respond to the cancer like an infection. But the cancer, of course, has to figure out a way to survive. That's what its goal is to make more of itself. And as part of that toolbox of skills that a cancer needs to achieve its own nefarious goal is really the ability to evade the immune system. And this has been a very difficult toolbox for us to defeat until very recently, when we have come up with very specific medicines that have removed the brakes that the immune system usually uses to regulate itself, to allow it to go a little bit further than normal physiology would allow, to overcome the obstacles that the cancer is throwing up, removing these cloaks so that it can see the cancer as being something foreign or dangerous. Now, in some patients, immunotherapy by itself may produce extraordinary results. But in other cases, we definitely need to combine it with other forms of directly killing the cancer, like radiation therapy, like Dr. Chan is an expert in, or targeted therapies or chemotherapies. But I think what we're now very excited about is that we are not seeing short-lived responses from immunotherapy. We are seeing durable responses, which last years, if not lifetimes. And that is the promise that we hope to achieve, that turning cancer from something that the patient is dying from into something that they live with or live without.
So Dr. Chan, as we heard a little bit earlier, some patients respond remarkably well and others not so well. This has been an area of your research and interest. Tell us a little bit about what you found about why this is the case.
So this started because Dr. Wolchuk, in the beginning days of immunotherapy, when he and others had first started putting these new medicines into patients to try to control the disease, noticed that in some cases, patients would do very, very well. And in some cases, patients who ordinarily would have disease spread everywhere and would usually not do well, have sometimes had disease control 5, 10, beyond 10 years. And that this was not just simply just a few patients now, but are actually quite a few patients over time. And this wasn't realized because, you don't see the long-term responses until you get to that point in time. So it took a while, but not everybody responded the same. So we work together now to use very effective genome sequencing of tumors, asking the question of what is it about the changes in certain patients' DNA, in certain patients' blueprint of their cancer cells that are altered that allow patients to achieve these great responses, and conversely, why some patients don't respond. And therein lies a scaffold, a rationale for figuring out what combinations one should use later on in clinical trials going forward, and also what specific compounds a given patient should get.
Dr. Brenchens, your work is really quite different than Dr. Chan and Dr. Wolchuk. You're taking these immune cells out and teaching them, showing them what the bad guy looks like. Tell us a little bit about how it's different and how it works.
Sure. Unlike the presumption that my colleagues make that there is in fact a revivable anti-tumor response, immune response, which is certainly the case, we 15 years ago went from the assumption that that response may not exist, and so we would want to create that response. And creating that response can be done through a re-education process of a patient's own immune cells. Now, as has been brought up, cancer cells are sometimes not recognized as being foreign at all, or if they are recognized as being foreign, they create an environment around it which inhibits the immune system from properly targeting it. And so rather than take an approach of a vaccine or other approaches to modulate the immune system, we decided we were going to take that immune system, what we call T cells, out of the patient, and then we were going to genetically alter those cells and instruct those cells to recognize certain proteins on the surface of cancer cells.
And the T cells are one type of white blood cells that are part of the immune system.
That is correct. And these T cells are a very important aspect of the immune system. And they recognize, and we understand how they recognize foreign proteins, or how they know how to attack through a receptor, attack diseased cells or infected cells. And so based on that knowledge, we utilize other aspects of the immune system to create what we call artificial receptors, or chimeric artificial receptors, or CARs, that we can then have these immune cells express, and when they now go back into the body, they find and ferret out the cancer cells, and they're able to kill them as if they were foreign cells.
Dr. Wolchuk, let's just take a little bit of a half step back, if you will, to talk about the immune system kind of in general. Its job is to recognize, we call it self versus non-self. Things that belong to me and are okay, and we're going to leave them alone, and things that are not me, whether it's an organ transplant that comes from somebody else, or bacteria, viruses, and so forth. How does it do that? These are little markers on the surface of all our cells that the immune system looks for?
It's a great question. Might take a few hours to answer, but so
got a couple of minutes.
But you're entirely correct in summarizing that the basic job of the immune system is to distinguish self from not self. And the way that it does that is with very specific markers on its surface that recognize something as either resembling a normal body part or not. And there's actually a small gland inside all of our bodies, which when we were very young sat on top of our heart, called the thymus gland. And for most of us in the room, except for Dr. Chan, who's relatively younger, or at least looks like younger, the thymus gland kind of gets smaller over time and kind of goes away. But the thymus is the place in the body where the entire population of white blood cells, called T cells, learns what the normal body structures look like. The thymus is a very, very interesting organ because it can show almost any normal body parts, component pieces to the immune system. And so most of the white blood cells that recognize normal body parts have been eliminated from our immune system. And so that what you've got left are either some white blood cells that recognize normal body parts with relatively poor accuracy, and everything else, which recognizes bacteria, viruses, anything, or someone else's body parts. And so we're left with that conundrum of really, how does the immune system see the cancer cell, which for all the world looks like the normal cell that it came from, that the thymus told it, don't look this way. How does the immune system see that? And we now believe, based upon really Dr. Chan's team's work, that it's these genetic errors that happen as a result of the cancer forming, which just by accident make the cancer look different than the normal body part it was told to ignore, and therefore it gets a bit of a go signal from those genetic errors.
So, Dr. Chan, that's what we sometimes call the immune surveillance theory of cancer, that we may possibly be making cancer cells all the time, but usually the immune system sees it and destroys those cells, and every once in a while some will escape?
That is the prevailing thought, actually. But although, now that the field is maturing, I think there's going to be a lot of investigation exactly along those lines. But what Dr. Wolchuk was saying before was, during the development of a lot of these new compounds, like IPI and like PD-1, this largely sprang from the desire to take the brakes off of the immune system, right? It was difficult at that time to understand or to predict how a cancer cell would indeed respond. You're essentially adjusting a threshold of immune activation. So if the immune system ultimately has evolved over eons to attack bacteria that perhaps went into your arm when you cut yourself to cause an inflammation reaction. Well, it turns out that the immune system can also likely ferret out small changes. Remember, a cancer cell is 99.9999% similar to the human body, right? And so the reason why transplant, however, doesn't work between you and me or you and your neighbor, for instance, is due to those small, tiny differences. Very, very potent, right? Despite the vast amount of similarity. So the drugs that Dr. Walchuk and others have developed really take advantage of allowing the immune system to see those minute differences. Not such an easy thing to do.
Dr. Brinson, Dr. Chan mentioned taking the brakes off the immune system. That sounds like a good idea. Let's just let her rip.
Yeah, it's a
That's
terrible
not
idea.
necessarily such a great idea.
It's a good idea for cancer therapy. So the immune system has checks and balances. It wants to mount a response to something foreign, but is afraid that if it mounts too vigorous of a response, it will be bad for the subject at large. And one can appreciate that when one gets the flu, the night sweats and the fevers, et cetera, you're not being able to get out of bed, is not a consequence of the flu, it's a consequence of the immune system. And that means your immune system is working well. However, if it works too well, it can sometimes wind up throwing the baby out with the bathwater, as it were. 15 years ago, when Jed and I, and when we were all actually just starting out, trying to figure out what the immune system does against cancer, we didn't appreciate how many checks and balances there are. But if those checks and balances fall out of balance, you either don't have enough of an immune response or you can get autoimmunities. And the body actually very much, the immune system very well fine-tunes itself so that it doesn't overreact to anything. And that is great for the overall well-being of the body, but it's terrible when you can mount an immune response against the cancer and your body, because if it's a natural reflex, says, taps it down and suppresses it. And so the brakes, taking the brakes off, is in fact taking the brakes off is hitting the gas pedal and reversing that natural inclination of the immune system to not overreact. And now you're instructing it, it's okay to overreact in this particular setting.
And you mentioned autoimmunity, which is a malfunction, if you will, of the immune system, where it leads to rheumatoid arthritis,
Exactly.
lupus, MS.
So if that fine balance is a little out of balance, it can cause damage to the patient's own body, mediated by its own immune system that is misconstruing something as being foreign when in fact it isn't foreign.
What kinds of cancers has immunotherapy been shown to be effective against so far, and what are the hopes where it might actually go from here?
So I'm going to address that, but I also want to expand a little bit on what Dr. Brenchen just said. I think it's important that we are expressing a lot of optimism for this field, but I think that there is a downside to letting it rip, right? And that we have seen side effects from immunotherapy that look very much like these common autoimmune diseases, like rheumatoid arthritis or Crohn's disease in particular, where they're very temporary and we can treat them. But I think it shows us the very tight balance we have to have between autoimmune health and autoimmune disease and this very, very double-edged sword of immunotherapy. So we've become very adept at treating these side effects, but we're aware that the immune system is a very powerful organ. And it can treat more cancers than melanoma, which is what we've spoken about so far. The same medicines that we have seen have activity in melanoma have also had efficacy and have led to drug approvals in non-small cell lung cancer, which is actually the most common cause of cancer death in North America right now. And also, we've seen activity in head and neck cancer, in bladder cancer, in Hodgkin's lymphoma, and in kidney cancer as well. What ties several of those diseases together is that they all have a relatively high number of genetic errors in them. Many, many mutations or changes in the blueprint. And in melanoma, that's frequently a result of sun exposure. For the others, they are all tied together by tobacco exposure. And so this gets to the kind of framework that we've been discussing, where the more different the cancer cell looks than the normal cell, the more likely it is that the immune system will see this as being a rational target, and for us to be able to help those patients with the medicines that sort of step on the gas a little bit. When we have more challenges with sort of not recognizing the cancer as being something foreign, then the type of therapies that Dr. Brenchen's team has pioneered, which puts immune cells in university for a little while outside the body, teaches them what to recognize, and then puts them back in, then that can become a very thoughtful intervention.
So ironically, the more mutations a cancer cell has, which normally you would think would be a bad thing, makes it more aggressive and nastier, that's actually better as far as immunotherapy goes because it looks more different.
Right. So in general, how a lot of targeted therapies work is, you would study the biology, and you would pick a drug that would inhibit one enzyme that's messed up, that's screwed up, and you try to, and if that particular change was required for the tumor cell to keep on growing, then that would be a success, right? Like Placebo or EGFR inhibitors that are for lung cancer. Now, it turns out that if your tumor is very, very complicated and you have thousands of mutations, there are many, many solutions for the cancer cell to try to get around this new block that you've instilled. It turns out that immunotherapies, because of the nature of how these immune cells work, is that they target these new non-self changes, right? So it's actually better for immunotherapy that the more changes you have. Now, I'll just simply add, based on what Dr. Wolchuk was saying, is that one of our first findings is that a new drug that just got approved very recently, a few months ago, called anti-PD-1, works very, very well for smoking-related tumors. So, and it turns out that those are exactly the ones that Dr. Wolchuk had mentioned, where people saw initial responses: lung cancer, head and neck cancer, bladder cancer. These are all the smoking-related tumors. It turns out that is so because the smoking mutations, the little changes in the DNA that smoking causes, is just about 3 to 4% more likely per change than a non-smoking-related change to cause something that the immune system would see. You multiply this by 1,000, and suddenly you see a big propensity for smoking-related cancers to respond.
That doesn't mean smoke, because you'll get a cancer that's
So as a physician, I still recommend don't smoke, all right? But, in the past, smoking-related tumors were horrible, all right? They were the ones that were more aggressive and much more difficult to treat. I think the work here demonstrates a paradigm where this class of drugs can be very useful for smoking-related tumors, which, historically have been very hard to treat.
So, Dr. Wolchuk, how do these medications, particularly, for example, the melanoma ones that we're talking about, how do they take the brakes off the immune system? How does a medication do that?
It's a really important question. So most of these medicines work by blocking proteins that normally keep the immune system from becoming overactivated. So an analogy that we often use is that starting an immune response is akin to starting your car. And it takes multiple steps to start your car. The key has to be inserted in the ignition and it needs to turn. But that's not enough to get the car moving. There needs to be the foot on the accelerator. And those are two distinct steps that need to happen between an immune cell and a cell that it wants to recognize. Once that happens and the car gets moving, then you need to have a brake. And the brakes that we're talking about are proteins that have not such easy names to remember. One of them is called CTLA4, and the other one is called PD-1. And we know that these braking proteins keep the immune system from achieving a state of hyperactivation. And we know this because if you breed a mouse to lack one of these proteins, the mouse will actually die when it's 3 weeks old with white blood cells in every organ in its body. So sort of full body autoimmune disease. So here you see what complete absence of one of these braking molecules can do. So what we've done is we have worked in teams with pharmaceutical companies to make molecules that block these brakes, but temporarily. Maybe for just a few weeks, not for a lifetime, so that the immune cells can make more of themselves and become more activated than they otherwise would, but not forever. And these proteins are called monoclonal antibodies, which are a kind of immune therapy that are actually used to treat many different diseases. Some commonly used medicines to treat lymphoma and breast cancer are actually monoclonal antibodies that block in lymphoma a molecule called CD20, and in breast cancer a molecule that's very well known called HER2. But here we're using these same antibody-like drugs to block immune proteins that keep the immune system at bay.
So, Dr. Brenchens, let's talk about your approach here, taking these cells out and re-educating them, or as Dr. Wolchuk said, send them to university. I thought it was like showing them the 10 most wanted poster and saying,
it's about as expensive as sending them to university currently. To carry the analogy a little further, you can actually give these cells advanced degrees as well to help educate them how to protect themselves from just that same issue. You can imagine, I take out a cell and I re-educate it to recognize a tumor. So if we start at the bottom panel to everyone's left, you can see that we take cells out, immune cells, white blood cells, out of the patient, and we isolate the T cells from that. This is all done in a super specialized good manufacturing process, or GMP facility, that we have at Sloan Kettering. And Sloan is somewhat unique in that regard because there's not many institutions that have facilities to carry out this very complex procedure, which is markedly simplified on this cartoon. We introduce the gene through a virus. So we take a virus that doesn't cause infections, but allows for the gene that encodes for the receptor to be inserted into the immune cell. The immune cell then expresses that receptor. As you can see, that's the yellow S-like sign that's on the surface. That cell is now capable of recognizing a tumor cell. And so we grow those up and expand them, and then we bring the patient back and we give them back their now modified cells. Now, that's a great concept, and it was fully valid 15 years ago when we started working on this and when we were much less knowledgeable about what the tumor could do to avoid being killed by those cells. And as Dr. Wolchuk pointed out, if those cells are expressing those ligands, those breaks, then we can make all the cells we want in the world. Those cells are still going to be broke or slowed down or stopped in their tracks by a wily tumor cell that knows that even if we try to engineer it to recognize, that's not sufficient. And so one can then, if we want to carry the analogy further, one can see our cells kind of as bombers, but those bombers need fighter escorts. And so this is where, after years of us working in separate aspects of the immunotherapy, we almost now start to see a convergence, because I can imagine I make the cells that recognize the tumor, which may or may not be recognized, and I infuse them with Jed's help. We can now have those cells protected from those breaks, and now they can go ahead and do the business that they want to. It's actually very rewarding to see that as we learn more about it, we kind of were seeing the same problem from different aspects of it, and that what they're working on with antibodies and checkpoint blockade and what we work on with cell therapy is beautifully complementary. And not that I can't fall asleep at night, but the thought of combining those two and seeing what more it can do for patients is really extremely exciting, because on paper, at least, this should be a fantastic combination.
Is there anything that an individual can do to bolster their immune system, short of obviously going through taking these medications or whatever, that might actually improve their chances of dodging cancer?
That's a question that I get asked every week about 50 times in clinic. And so I think that it's a logical question, because we want to know what we can take back into our own control. And so the truth is that there really isn't anything that has been scientifically proven that you can eat or drink or rub behind your ears that will absolutely improve your immune response to cancer. I think what I usually recommend is that people, first of all, eat a balanced diet, not in any one direction or another, that people do things that they enjoy. I think that there is a long-standing study of psychologic function, depression, happiness, and immune function. Get a good amount of sleep, do things that you enjoy. And I think that there have been some studies that correlate those, but I don't think yet there's something that we can tell you to run down the street and buy from a health food store that will absolutely improve your immune response to cancer.
We've heard recently about using the polio virus to, again, unleash the immune system in the case of brain tumors. Talk to me about that.
I think there are a few reasons. One is sort of the Coley hypothesis, right? That but by introducing a bacteria or a virus into the body, you're going to unleash this immune response that's rather nonspecific. But some of these viruses that are being used are specifically called oncolytic viruses, meaning they can actually kill cancer cells directly. We are studying some of those here. There is work looking at various kinds of herpes virus, one that's in late-stage clinical trials. There's measles studies that were done at the Mayo Clinic. There was the work done with polio. And so we think of these viral strategies as being combination approaches. They may actually kill some tumor cells, but they also stimulate the immune system in a nonspecific way. And so I think that it is another reason to believe that immunotherapy needs to be studied much more closely in the future.
Let's talk about how you decide who may be appropriate for immunotherapy.
In melanoma, we believe that immunotherapy absolutely needs to be a part of every single patient's treatment because there are numerous approved immunotherapies for this disease which have all demonstrated an ability to extend survival, not just induce short regressions, but lengthen the time that people live in a meaningful way. We used to believe that immunotherapy was an alternative to standard cancer therapy. It was an investigational option. Now I think we recognize that immunotherapy is an additional fundamental standard pillar of cancer therapy. It no longer is just an investigational option or an alternative. It's something that has bona fide activity on its own, and I believe that in combination with other approaches, the future will be quite bright.
We thank you very much for being here this evening, and we've given you hope for the future. Thank you.